So I'm Chun-Hong Ma from Shandong University, and I'm an immunologist, so more specific topics. And so happy to have this chance to share with you about our recent work, which focuses on liver NK cells and HCC. So next one, please. So NK cells, as we all know, is the killer cells, is a very efficient killer cells, which can kill the tumor cells directly by secreted perforin and granzyme. And also NK cells can secrete the cytokines, for example, the interferon gamma, to play an immune regulation role. So next, please. And so there are a lot of evidence shows the negative correlation between NK cells and the incidence rate of cancers. And here show the Lancet report on 2000. You can see both in men and women, the higher residence of NK cells correlates with the lower incidence of cancers. So next, please. And also, many reports shows that the NK cells, both the number and the function of NK cells, decreased in the tumor microenvironment, and NK cells display as increased inhibitory receptors such as the PD-1, TIM3, and NKG2A, and also the decreased activity receptors, for example, the NKG2D and the NCR1. So next, please. And so NK cells in the liver present about 20 to 30% of the total hepatic lymphocytes, which is about 5 times higher than the spleen and the peripheral blood, and also the unusual high frequency of the hepatic NK cells coincident with the critical roles of NK cells during liver tumorigenesis. And you can see the NK cells incidence decreased in the tumor, but not in the non-tumor tissues. So next. And now about the notably over about 5 or 10 years, accumulated data demonstrate that the heterogeneity of liver NK cells in the liver, at least 2 NK populations. One is the next, the CNK, we call the conventional NK cells, and the other is we call the resident NK cells. The conventional NK cells can circulate in the blood, but for the liver resident NK cells, which will develop in the liver and also resident in the liver, but both of the 2 subsites can express the mark of NK cells, which is NK1.1 and NKp46, but they are different with the marker, what is the CD49A and the CD49B. Next. So that is the mouse. And in human, now people have demonstrated that the liver NK cells also has 2 subsets, which one is the CD56 bright, we call the resident NK cells. The other is the CD46 dim, which is the conventional NK cells. So both the liver resident NK cells, the CNK cells, they also have the mark of the NKp46. So in the reports before we discover the liver resident NK cells, all the reports may be overdue for the whole 2 subsets. So our question is, what's the role of the liver resident NK cells? Does the liver and resident NK cells seem like the CNK cells? So next. To address that, we built a marine model of orthoptical HCC models, and as you see here, we can see the liver resident NK cells and the CNK cells in the liver and the tumor site of the mouse liver. And as the report, both the number and the percentage of NK cells, the total NK cells and the CNK cells is decreased. And also we saw the decrease of the liver resident NK cells. Very interestingly, although the number of the liver resident cells decreased, but the percentage increased in the tumor. So what's the reason we are now still studying? Next. Then we come to analyze the function, and so we saw, as reported, the function of the CNK cells decreased in tumor compared with the paratumor, and also it's similarly for the liver resident NK cells. You can see the decreased expression of the actin receptor and the interferon gamma, which is the cytokines. So the liver resident NK cells and the CNK cells has decreased function in the tumor site. Next. So that is the mouse data, and then we come to the human data. We do the overall multiple fluorescence staining to see what happened in our human HCC, and also we do the flow cytometry, and you can see both the number of the liver resident NK cells and the CNK cells decreased, and it shows a net correlation with the survival of the patients. And then next. So we now detected that the number and the percentage of the liver resident cells and the CNK cells are decreased. Then so why? Next. So as we all know, for the T cells, the immune checkpoints, including the PD-1, CTLA-4, and TIM3, are upregulated in the T cells. And next. And this has been used as the target for the tumor therapies. And next. And this is also true for the NK cells. NK cells can express much higher expression of the immune checkpoint, and blocking immune checkpoint can reverse the exhaustion of NK cells. Next. So we come to compare the immune checkpoint expression in the tumor infiltrating NK cells within our human samples. And as you can see, both all four molecules, including the TIM3, PD-1, LAG3, and TIGIT, are increased in the tumor site compared with the paratumor. And among all the four molecules, we see the highest one is the TIM3. And also we can see that TIM3 is not only increased in the CNK, but also in the liver and NK cells. And next. And then we also use our multiple fluorescence staining to see much more patients' samples. And you can see in so many patients, the TIM3 are increased in the liver resident NK cells and CNK cells. And you can see a negative correlation with the patient survival. Next. And then we confirm this data with our mouse models. Next. So what is TIM3? TIM3 is a memory molecule, which is very like as the PD-1, but unlikely, TIM3 not only expressed in T cells, but also expressed in many different kind of immune cells, including the macrophage and NK cells. And next. And also, somebody showed that the TIM3 is high expressed in the mouse with the resistant to the PD-1 therapies. And when you combine with the TIM3 and PD-1, it can show you a much better effects. Then next. So what's the function of TIM3 in this TRNK cells? Next. So we prepare the knockout mouse with the TIM3. And as you show here, when you knock out TIM3, the NK cells function are decreased. And this is both for the CNK cells and the liver resident NK cells. Next. Then we come to the tumor mouse model with the HCC, and then we isolated the NK cells from the tumor and blocking the TIM3 with the blocking antibodies. And as you see here, when you block TIM3, the NK cells functions are increased both for the total NK, the CNK, and the liver resident NK cells. Then next, please. Also, we confirm our data with the human samples with the human antibody against TIM3. Next. So we now know the TIM3 is responsible for the dysfunction of NK cells. So how it can work. So unlike the PD-1, TIM3 has different kind of ligands. At least we now know 4 kind of ligands, including the galectin-9, CCAM1, HMGB1, and phosphorin serines, we call the PS. Next, please. So we detect all these known ligands of TIM3 in our human samples, and you can see the phosphorylation of the TIM3 is the highest expressed in the tumor cells and the NK cells. And also, when you treat the NK cells with the PS, the TIM3 can be phosphorylated, means the PS is the ligand of the TIM3 in the tumor infiltrating NK cells. Next. And then we treat our NK cells with the PS, both for the wild-type mouse and TIM3 knockout mouse. And you can see very clearly with the wild-type mouse that the PS can suppress the NK cells function. But without TIM3, this function disappears. That means the PS inhibits the function of the TIM3 of the NK cells while binding with the TIM3. So next, please. And then we come to the signal pathway. And so I will come more briefly. Finally, we find the interaction protein in the cytoplasmic. We can see the TIM3 can bind with the P85 and interfere with the PI3K pathway. Next. And then we come to the more detailed structures. So we do many mutants. One is the mutant in the extracellular domain, which is the binding motif of the PS, and also in the cytoplasmic domain, which is the phosphorylation of the TIM3, including the Y265, Y272, and both mutations in the 2 sites. And with the iPSC, we can see that both the site is important for the PS binding with the TIM3. And next, please. And next. So with multiple assays, we finally can see that TIM3 can interact with the P85 and inhibits the combination of the P85 with the P110. And next. And then we detect the downstream signals and also show this the signal pathway. So next, please. Much slow. So maybe next one, and I will ignore this detail. Yeah, and also this, so with a lot of experiments, we clearly show that the PS can binding with the TIM3 and leading to the signal pathway to inhibit the PI3K, AKT, mTOR in the tumor infiltrating NK cells. And lastly, we come to assay if the TIM3 can be used as the immunotherapy. Next. So we prepare 2 kind of animal models. One is the TIM3 coming from the TIM3 knockout mouse and the wild-type mouse, and transfer to the tumor with to the mouse with tumor. And you can see clearly the TIM3 knockout mouse NK cells can significantly inhibit the tumor growth. And also we confirm this result with the TIM3 lentivirus, and so this shRNA. So TIM3 can suppress NK cells and then promote the tumor growth. And next. So since TIM3 can be expressed both the so for the last lay, we do the survival assay of the mouse. You can see here when you use the TIM3 antibody, the survival of the mouse can be lasted for longer. But when you delete the NK cells with the antibody anti-GM1, then you can see the effects is inhibited. That means TIM3 can inhibit the tumor growth in an NK-dependent manners. So next one. So here is our working model. In the tumor microenvironment, the tumor cells can express the PS on the cell surface. And this PS can bind with the TIM3, and the binding will lead into the exhaustion of the liver-resident NK cells and the CNK cells, and then make the tumor cells escape from the immune attack. And for the molecular mechanism, the binding of the TIM3 with its ligand PS can leading to the interaction of the P85 with the TIM3 cytoplasmic domain, and then this P110 and the P85 can be broken down and suppress the PI3K mTOR pathway to suppress the NK cells function. So that is our recent work. And so lastly, okay, I will thanks for my students and for my cooperations. And so that's my talk. And I'm be happy to take some questions.
So, if you look at the development of tumor, you have inflammation, and this is increase the possibility for tumor to develop. So it's possible that some of the immune cells may even, feed the tumor.
Yeah.
So and K cell could be one of the candidates. So you showed, cells within the tumor, and you say they don't believe right. They don't behave right.
Mm-hmm.
Did you, let's say, did you try to get, to extract RNA from those cells and check what they produce? It may be that they just do the opposite, what you think. They just feed the tumor. Okay, so they just, if you eliminate them, it's possible that the tumor die.
So actually, if we eliminate the NK cells or deplete the NK cells, the tumor will be grow more rapidly.
Okay. This is in the mouse. Is it in the mouse, or did you really check tumor tissue in the human tumor tissue?
We did it in the mouse, also in the tissues.
So if you take tissue from human tumor and you check as you did, you check how many cells are there, and then you extract RNA, for example, and see what do they produce.
Yeah,
we don't know. We don't know. We don't know what are the growing factors for the tumor cells. They just, they have, we have serum, and we have a lot of all kind of factors, and those cells may also do the same thing. It's just the opposite way to think, the opposite way to think how a tumor, stay alive and grow.
Yeah, we do the tumor tissues from the patients and isolate the NK cells, and then assay the NK cells mRNA or the profile, the expression profile. They can produce a lot of cytokines.
Right. One of them can, the surviving factor for the tumor cell.
I don't think so. Not.
No?
Yeah.
Okay.
Yeah.
Good. Just an hypothesis.
Yeah.
Questions?
Yeah, I can ask a question. I mean, Dr. Ma, very nice work, really molecular details. So I miss a little bit the therapeutic points. I mean, you would like basically to try to reconstitute the NK cell function with an anti-TIM3 antibodies?
So the antibodies can block in the NK cells and also the T cells, since the TIM3 express in different kind of immune cells. So this is I think it's more better than PD-1, since PD-1 is mostly expressed on T cells. TIM3 can express both NK and T.
And if I can ask a short question again, I'm curious to know whether you test also the NK cell function in liver transplanted patients that are under immunosuppression, and whether you find the NK cells that are also, let's say, are abolishing or diminishing their function. Have you ever tried on the patient, let's say, in some data, because, I mean, there are many patients now that are developing hepatocellular carcinoma metastases, for example. I'm just wondering whether you have tested the immune, let's say, environment of the liver in liver transplanted patients.
We didn't try the liver transplantation. We only get the tumor samples, but maybe you are right. Yeah, we tumor.
Okay. No questions anymore? Thank you. Thank you very much, Dr. Ma. Thank you for sharing